Autophagy plays an indispensable role in maintaining stem cell longevity, acting as a guardian of their function, vitality, and regenerative potential. Stem cells are the body's reservoir for repair and renewal. They replenish tissues, heal injuries, and maintain organ integrity throughout life. However, as we age, the regenerative capacity of stem cells gradually declines, leading to thinner skin, slower wound healing, weakened muscles, brittle bones, and a general reduction in tissue resilience. One of the lesser-known but profoundly important factors behind this decline is the age-related drop in autophagic activity. In youthful tissues, autophagy helps stem cells remain in a state of readiness by cleaning out damaged proteins, oxidized lipids, and dysfunctional mitochondria that would otherwise interfere with their energy metabolism and genetic stability. This internal maintenance ensures that stem cells do not accumulate harmful cellular waste, which could push them into senescence or trigger faulty differentiation. In essence, autophagy preserves the youthfulness of stem cells by preventing toxic buildup that would impair their ability to divide, migrate, and generate new tissue. Mitochondrial health is particularly critical in stem cells, as their energy demands shift dramatically when they transition from a dormant state to an active, regenerating one. Autophagy, specifically mitophagy, helps ensure that only the fittest, most efficient mitochondria remain, enabling a clean, responsive switch between metabolic modes. This efficiency allows stem cells to respond more quickly and effectively to injuries or cellular turnover needs in organs such as skin, muscle, liver, and bone. Without autophagy, damaged mitochondria begin to accumulate in these cells, reducing their ability to adapt and reproduce. Over time, this contributes to the sluggish regeneration and fragility often seen in aging tissues. Another critical aspect is genomic stability. Stem cells, by their nature, must maintain pristine DNA to avoid passing mutations to future generations of cells. Autophagy helps buffer these cells against DNA damage by reducing oxidative stress and removing organelles or proteins that produce genotoxic byproducts. When autophagy is impaired, oxidative stress rises, DNA repair systems are overwhelmed, and the risk of stem cell depletion or mutation increases. This is one of the reasons autophagy is also being explored in the context of cancer prevention. Healthy stem cells are less likely to become the origin of tumors when autophagy is intact and functional. Interestingly, autophagy also plays a role in regulating the balance between stem cell quiescence, resting state, and activation. Quiescence is necessary to preserve the stem cell pool over time, preventing early exhaustion. But when tissue damage occurs or renewal is needed, autophagy helps fuel the transition into active regeneration. It provides internal nutrients and energy during times of metabolic stress, such as injury or nutrient scarcity, enabling stem cells to respond quickly without becoming depleted. This fine-tuned regulation keeps the regeneration system both stable and adaptable, key traits of a healthy, aging-resistant body. The benefits of this extend across numerous tissues. In skin, autophagy preserves epidermal stem cells, supporting firmness, barrier repair, and youthful texture. In muscle, it helps muscle stem cells, satellite cells, regenerate fibers after exercise or injury, maintaining strength and preventing atrophy. In bone, autophagy sustains the osteoprogenitor cells responsible for producing new bone tissue, supporting density, flexibility, and fracture resistance well into later life. Even in the blood and immune system, autophagy ensures the smooth turnover of hematopoietic stem cells, which generate all immune and red blood cells. Fortunately, this protective mechanism remains modifiable throughout life. Fasting, exercise, and certain polyphenol, like quercetin, curcumin, and resveratrol, are known to induce autophagy and improve stem cell function in both experimental and real-world settings. For example, exercise not only increases autophagy in muscle fibers, but also activates muscle stem cells, enhancing repair and regeneration. Intermittent fasting or time-restricted feeding can likewise stimulate autophagic activity in skin and bone progenitor cells, promoting more youthful tissue structure and function. In a broader sense, autophagy allows the body to preserve its regenerative capital to maintain the pool of stem cells that will be called upon throughout life. This preservation of potential is perhaps the most important anti-aging effect of all. Rather than attempting to reverse aging once it has progressed, supporting autophagy means aging more slowly in the first place, remaining more resilient, more responsive, and more capable of recovery at every stage of life. It's not a single solution, but a central lever of longevity that touches nearly every system of the body, beginning with the foundational blueprint for regeneration, the stem cell.